Hello, my name is Lindsay Bayrekohler, and this summer I worked with Brian Rudder, Peter Tomlinson, and my mentor, Dorivar Ruiz-Diaz, on this project, soil health parameters and early season nitrogen uptake in corn. Oh no.
Page down.
So a little bit of background. This summer, my project was actually just a small subset of a project that's being worked on, looking at agronomic value of soil health parameters. But that being said, let's get into some of the rationale behind my project. Recent developments in soil health testing have led to increased popularity and demand in attempting to use these tests to adjust fertility recommendations. But one problem is that data relating these soil health parameters to crop response is scarce. And what's the use in running all these new and expensive tests if we can't relate them back to make producer recommendations? So this project was looking into some select soil health parameters that could prove useful in making these recommendations. Some concise objectives would be to investigate correlations between soil health parameters and then to evaluate their relationship to nitrogen uptake in corn under field conditions. So getting into some methodology, nitrogen response trials were conducted at 15 site years across Kansas in 2017, 2018, and 2019. Pre-plant soil samples and early season plant samples were collected from control plots. So those that didn't receive the nitrogen treatment, which basically means that any nitrogen taken up by the crop, in this case corn, was already in the pool in the soil to begin with. But there were 4 control plots at each site, which gives us a total of 60 samples. Lab analysis consisted of routine fertility testing, so things such as soil organic matter, pH, phosphorus, potassium, and soil texture. Lab analysis also consisted of soil health parameters more specific to this study. So 24-hour CO2 respiration, which is an indicator of overall microbial activity, POXY or permanganate oxidizable carbon, which is just a fancy name for active carbon, shows early metabolized forms of the carbon, so those readily available for organisms within the soil. NAG or N-acetyl beta D-glucosaminidase, which is an enzyme that is a useful catalyst in the breakdown of complex carbon and nitrogen structures. And then ACE, autoclave citrate extractable soil protein, which is a measure of potentially mineralizable nitrogen, which means the nitrogen can go from organic to inorganic forms and become bioavailable. Statistical relationships were investigated using the Pearson correlation coefficient and evaluated at the 95% confidence level, with analyses completed in R and RStudio. So figure 1 shows distributions of general soils information from all sites included in the study. So soil texture up here, a lot of soils range around 20% sand, 50% silt, and 30% clay. A lot of the pHs fell between 5.5 and 6.5. And all the soil organic matter was between 3 and 4%, as well as a variety of concentrations in profile nitrogen, phosphorus, and potassium. This figure kind of just illustrates the variety of soil types we had in the study. So figure 2 shows distributions of soil health parameters measured in samples included in the study. And if you notice, this one is split into 2 groups. So the purple represents conventional tillage. So that was things that were more aggressive, like disc tilling was used at these sites. And then the green is something we deemed minimum tillage, which consists of strip tilling and or no-till practices. So just visually, you can kind of see that some of them have a separation between the 2 practices, but keep in mind that this isn't necessarily due to the tillage practices themselves. It's merely a representation of the soils we had in our study. So getting into correlation plots. These are also split by tillage. And just to help you get a better understanding of what you're looking at, the darker the purple, the higher the positive correlation, the darker the green, the higher the negative correlation. And white would be no correlation between the parameters. As you can see, minimum tillage has a lot clearer positive relationships between the parameters in many more, between many more of the parameters, noted by all that purple right in there. But something else that is important in these plots is the bottom row, which is looking at V6 nitrogen uptake. So we're going to look a little bit closer into a few of the parameters. Soil organic matter, which is outlined in the blue there, and ACE soil protein, which is outlined in the red. These are chosen simply because of the higher positive correlations seen, at least in minimum tillage. So this is early season nitrogen uptake versus soil organic matter. Now, soil organic matter is actually a parameter already used by KSU to make fertilizer recommendations. I believe about every 1% of soil organic matter is assumed to get 20 pounds of nitrogen per acre, at least in warm season crops. In cool season, I think it's about 10 pounds. But here you can clearly see, like represented in the correlation plots, that minimum tillage shows a much clearer relationship between soil organic matter and nitrogen uptake at V6, whereas the conventional till has almost no to very little relationship between the two. Here, between early season nitrogen uptake and ACE, we see almost the same thing. Minimum tillage has a significant relationship between ACE and the nitrogen uptake, whereas conventional tillage, the relationship is not significant. So some takeaways are that correlations between soil health parameters and early season nitrogen uptake appear to differ between tillage practices, and that soil organic matter and ACE testing show the highest positive correlation to early season nitrogen uptake in corn with minimum tillage practices. But this is not the be all end all. Because this was only focused on early season uptake, we only got a snapshot of nitrogen uptake and what was in the soil at that time the samples were taken. So later studies could focus on late season and harvest nitrogen uptake as well. And we could further investigate the relationship between soil organic matter and ACE soil protein in relation to nitrogen uptake in corn to kind of further the possibility of helping make these recommendations in the future. Any questions?
All right, thank you, Lindsay. Any questions for